One ring to rule them all, a very Middle-earth statement. And from the perspective of single atom physics, it's incredibly appropriate. Oh, behave, empowering, healing. It's physics, not physical. It's light, not matter. It's monoatomic, not diatomic. It's inverted symmetry, not chaos. It's anti-aging and supports longevity. Okay, so if I start to talk about the physics of the product, not the physical nature of the product, it's a light-based photoreduction of a single atom of iron coordinated to the antioxidants bound to the proteins, the royal jelly proteins. So this light-based chemistry is known as photophenton chemistry. It generates a reduced iron, and the iron reacts with hydrogen peroxide. This generates a compound known as a hydroxy radical, and that produces 1200 electron volts of energy in 1 nanosecond. So this hydroxy radical has the power to turn biological molecules back into CO2 and water. It is this ability that is utilized by the product Oh Behave, Empowering Healing. Developed a new scientific model in order to understand how these single atoms operate. There was a new science model developed in the 1930s called quantum physics, and this provided a non-intuitive way to understand the structure of hydrogen in various forms and how each one of those forms can occur. So I've developed a new approach which unites the four forces within the atom to understand the structure of the atom and its properties, identifying the origin of time within atomic structure, and utilizing the idea of a pinhole camera where the inside and outside are inverted to one another. So this model is called supersymmetry inversion. So as I mentioned before, it's light, not matter. So photons, or the electromagnetic force of light, can change the oxidation state of a single atom ion from Fe3 to Fe2.
This is what the royal jelly proteins look like under nanosite microscope. You can see this wave-like behavior. It's the quantum properties. So where is this light energy coming from? Well, it's laser light, obviously illuminating and exciting the atoms that are operating within the product. It's a very strange thing to consider where this biophoton energy originates from.
When I was looking under the microscope, I discovered some very unusual properties of this product, and I've really been trying to understand how it actually works. And because it's a single atom housed in the aromatic ring of these phenolics that are bound to the proteins from Manuka honey, it doesn't behave normally. It doesn't behave like matter. It actually behaves like light. And the light that's generated is dependent upon the isotopes that are produced. So with the reduced iron, you can get a reaction with hydrogen peroxide that generates this hydroxy radical. And because it's so quick, happening with a half-life of a nanosecond, it's very hard to detect. But this hydroxy radical actually supports apoptosis. Apoptosis is cell death, or pre-programmed cell death, and what we're finding is it's helping remove old cells from a person's body. So I went back to first principle. I looked at Einstein's equation E equals mc squared and rearranged it to make it c squared equals E divided by m. The reason for doing that was to try to comprehend how the electromagnetic field or force within the structure of an atom is arranged. We know about the right-hand rule of electromagnetism. And so by rearranging Einstein's equation to make it light first, c squared, and then looking at the E divided by M as a right-hand rule of electromagnetism, or the E electric field and the magnetic field at right angles to each other, I came up with a functionally useful model to try to understand the geometry of photons within the structure of s orbitals within the atom hydrogen. And this is kind of the way I've been exploring the electromagnetic force within atoms, functionally looking at the structure. The atom is a single atom. It's known as a monoatomic, not like an oxygen atom of O2 or two atoms bound together in a bond with a bond. So this doesn't have any bonds. This is located in a quantumly, or a Faraday cage, you could assume, with the pi electron ring in the aromatic ring, providing a quantum environment for isotopes to do their thing. The single atoms, as you can see from some of the videos I've just shown you, actually behave in a very strange way, and they have unusual properties. What I've discovered or stumbled upon was a model whereby hydrogen being added into an atom through a particular cross-based geometry where positron and electron pairs occur, we can understand the idea of, well, there's no mass and no charge in a photon of light, and that provides the ability for the photon, given a particular wavelength, to quantum tunnel. So hydrogen has this unique feature of being able to be two photons of light because of that geometry. And what we're seeing is that these single atoms housed in the ring can receive hydrogen and then deliver hydrogen back out again. So it's really understanding hydrogen biology and the functional benefits that that can provide that is a big kind of step away from carbon-based biology that's taught in biochemistry. So I've had to really look at alternative ways of exploring isotope physics within biology, and this is really opening up a new doorway into human biology and understanding light within the structure of atoms and how that's being utilized in communication with biophotons.
The ability to stimulate photophoton chemistry and the generation of biophotons within co-behave empowering healing all comes down to some really interesting nanospeed second physics that's happening within the product. The release of the biophotons is really the release of the electromagnetic field within the structure of the atom itself. How this actually occurs is very strange. What is happening is that hydrogen is being added to the atom housed in the aromatic ring system, and that atom all of a sudden becomes unstable. It becomes an isotope, and that isotope decay is what's releasing the biophoton out of the single atom system housed in the aromatic ring. So this photophoton system is connected directly to putting hydrogen into the atom.
So it's inverted symmetry. What does this mean? It really is a different way of looking at atom structure, looking at stability of an atom based on the inverted symmetry through the single point of the nucleus, or the singularity. So it's a different model. It provides context for isotope physics and atomic decay based on the stabilization of the atom. If the number of electrons and positrons are equal and the number of neutrons and protons are equal, then the atom will be stable. So each isotope has a different time of its half-life, and that really provides information about frequency of operation, and it's like a radio station tuning to a different frequency. So what we've found is that we've got this high energy radical system that's present in Manuka honey that produces hydroxy radicals that is supporting health and well-being. That's the complete opposite of what the literature suggests. And because of such a paradoxical nature of this insight, this understanding, it goes completely against the mainstream. So what we've found is that there is a hydroxylation process that uses the hydroxy radicals in the pollen grain, and the pollen grain generates the methylglycol dihydroxy acetone as part of this pollen germination process that generates CO2 and water. And as the pollen explodes within the honey, it releases the polymeric materials, which then react with the proteins. So we have a very interesting system in operation that involves the utilization of the natural processes that are involved in fertilization within the Manuka flower itself. And the modification of lysines on the royal jelly proteins in the Manuka honey generate CEL, particular adducts, MGO adducts on lysines, and they are very similar to acetylation events that are happening on histone proteins. And what we've found is that the modifications cause a inhibition of this HDAC enzyme involved in epigenetic control of gene expression, and that's been identified by others as a way of controlling cancer as well as having anti-inflammatory properties. So there are a number of different regulatory pathways that are available. The technology is used as a topical cosmetic spray, but it's also used in wound care activities and is showing benefits in a range of other modalities or indications in early kind of clinical development studies and clinical case studies. And that's including in dementia and bipolar depression, PTSD. So this is a new technology. It is made from food that is utilized topically to stimulate regenerative healing. A whole wide range of benefits have been observed, and functional outcomes include accelerated wound healing, improved levels of vitality, and this is really supporting the anti-aging benefits and helping people live healthier for longer. Here are some testimonials from people. The product's showing some very interesting properties.
People talk about saying, oh, my heart said buy this, or, you know, my soul said buy this.
I just feel always clear energy.
Yeah.
Yep. And good with my decision making, feel sharp.
Yeah.
I spray it personally. I spray it once a day in the morning.
Yeah.
Rub it in on the chest.
Yeah.
And have been doing that for a good 12 months now. Yeah. And that works real well for me. Just to give you some feedback.
I did use the nasal spray myself.
Yep.
And that was amazing.
Yeah, what I can say is I applied the spray every day. I could only manage to do it like 5 times a day. But the night when I came home, one night when I came home after the operation, I applied this and it actually took the pain away.
For the continuation of the development of the intellectual property and this new revolutionary apitherapy that we've made from Manuka honey that supports health and longevity. Thank you.